Today, we're going to dive into the incredible story of how biology and computer science came together to decode the very code of life itself. It's a journey that takes us from punch cards all the way to artificial intelligence.
So let's just picture this for a second. Back in the 1960s, if you wanted to look up a protein sequence, you didn't log onto a server. You pulled a book off a shelf. That's right, the first ever protein database was an actual book. To give you an idea of the scale, the first protein ever sequenced, insulin, had only 51 amino acids. It was data you could literally hold in your hands.
Okay, now let's jump to today. The data in just one person's genome. We're talking about 3 billion base pairs. We've gone from a single book to what would be an entire library full of information for just one person. The change in scale is it's astronomical. So that's the big question, right? How in the world did we get from a single book to this absolute flood of data? And, maybe more importantly, what do you do when the book of life starts growing way faster than we can possibly turn the pages?
To really get our heads around this, we've got to go back to the beginning. We need to talk about the pioneers who first figured out the alphabet of biology and then had to teach it to a machine.
So the first thing you need to know is this core concept called the central dogma. Think of it like this. Your DNA is the master blueprint. That blueprint gets copied or transcribed into RNA, which is kind of like a daily work order. Then that work order gets read or translated to build proteins. And the proteins? They're the tiny machines that do, well, pretty much everything in our bodies.
And this is where our first hero enters the story, Margaret Dayhoff. She's often called the mother of bioinformatics, and for good reason. Back in the 60s, she had this incredible foresight to realize that the only way we were ever going to understand these huge biological sequences was with computers.
But the challenge she faced was so practical. I mean, look at this. They weren't typing on keyboards, they were feeding stacks of punch cards into giant machines. The old way of writing out amino acids was just too clunky, so Dayhoff's genius move was to simplify. She created a one-letter code for each amino acid. And get this, it was so elegant and effective, it's the exact same system we still use today.
So Dayhoff really laid the groundwork, but it was one single, massive event that was about to take this niche field and blast it onto the world stage, opening the data floodgates for good.
And that event was the Human Genome Project. It kicked off in 1990 as this huge public moonshot mission, but things got really intense in 1998. A private company called Cellular jumped into the ring with new computational tricks, and suddenly this scientific marathon turned into an all-out sprint. That competition just accelerated everything.
You know, the project's real legacy wasn't just the final map of our genes. It was the fact that it created a data problem so enormous it forced biology and computer science to truly merge. It demanded new software, new databases, a whole new way of seeing biology as an information science. This was the moment modern bioinformatics was really born.
So the race was won, the genome was sequenced. Awesome, right? Well, it kind of created a new, even bigger problem. We basically became victims of our own success. We got so good at reading the book of life that we were suddenly generating data way faster than we could analyze it.
This chart shows you the problem in a nutshell. For decades, we had Moore's law, right? That's the bar on the left. Our computing power was doubling every couple of years. Pretty good. But then look at the bar on the right. That's genomic data. Around 2004, it just went vertical, completely blowing past Moore's law. Our ability to create data just left our ability to compute it in the dust.
And it gets even crazier because the data isn't just one simple thing. It's got all these layers, what scientists call the omics stack. You start with genomics, the DNA, that's the master blueprint, then transcriptomics, the RNA, which shows what parts are active, then proteomics, the proteins, which are the actual workers, and finally metabolomics, which is the output, the actual activity. Each layer is just another mountain of data to climb.
So how does all this stuff connect? Well, that's where the metabolome comes in. I love this phrase for it, the omics superglue. It's called that because it's the closest thing to what's actually happening in your body, your observable traits. It's the final result of all those other layers working together. Okay, so you're facing this multi-layered, out-of-control data flood. You can't just wait for computers to get faster. The only option was to get smarter. Researchers had to invent totally new ways to search through all this information.
The old way was what you'd call brute force. It's like going into that giant library and reading every single page of every single book to find what you're looking for. But the new approach, compressive search, is a game changer. It's built on this key insight that biological data has a ton of redundancy. So instead of looking everywhere, it finds the patterns and only checks the most relevant parts of the library.
So why on earth does that work? It's because biological data isn't random noise. It actually has a hidden structure. This table shows a measurement called fractal dimension, which is just a fancy way of saying how complex or messy something is. And look at the numbers for biological data. They're incredibly low. That means it's super structured and predictable. Evolution has kind of forced life into a very small, non-random corner of what's possible, and these smart algorithms are designed to exploit that.
And all of these smarter algorithms, well, they set the stage for the revolution we're in right now. Once we could handle these massive structured datasets, we could finally bring in the heavy hitter, artificial intelligence. We're now moving from just reading the code to actually predicting what it's going to do.
And trust me, this is not just some academic exercise anymore. We are talking about serious investment. The US market for AI and biotech is expected to be over $2 billion. That tells you everything you need to know. This is a fundamental shift in how we approach medicine.
AI is being plugged into pretty much every part of drug discovery now. It's helping us find new targets for drugs in our genes, it's designing brand new medicines from scratch, and it's making sense of all those different omics layers. It's all pushing us toward that ultimate goal, personalized medicine, the right drug for the right person at the right time.
So where does this all lead? Well, one of the most mind-blowing ideas out there is the digital cell. I mean, just imagine it. A perfect virtual model of a living cell running on a computer. You could test new drugs on it, simulate diseases, and see what happens, all before you ever have to do a single experiment in a real lab.
But you know, the key takeaway here isn't about any single piece of software. It's about a way of thinking, systems thinking. To succeed now, you need biologists, data scientists, and engineers all working together, building these integrated platforms. We have come a very long way from the lone pioneer with her punch card.
And that brings us to a final thought to leave you with. For years, the story was that biology was generating data too fast for our computers to keep up. But with AI, that's changing. We're catching up. So let's flip the question on its head. What happens if biological discovery actually starts to accelerate at the same exponential pace as computing? Just think about that. What becomes possible then?